Thanks everyone for joining and welcome to this webinar in our PD series of webinars. As James said, I'm the Chief Scientific and Medical Officer at IXICO. We are an imaging CRO specializing in neuroscience and have supported CNS clinical trials for over 2 decades. I'm very excited about today's topic and today's discussion, where over the next hour or so we'll be looking at established as well as emerging application of imaging in PD clinical trials. I would say we're really at an inflection point in PD drug development at this point in time, where we see a wave of candidate disease-modifying therapies that entering clinical trials that are targeting underlying biology as opposed to focusing on symptomatic controls. We see alpha-synuclein aggregation, lysosomal dysfunction, dopamine pathway integrity, all of those underlying mechanisms being targeted by different drugs in development. We also see an increasing shift from clinical diagnosis to biological staging, which really helps us define in a more granular and detailed manner who has the disease based on what's happening at the molecular level, as opposed to looking at the symptoms. And imaging can be central to all of that. It is one of the key tools that can help put a patient on that biological map or that biological understanding of the disease, which is why I'm so excited to hear from our 2 speakers today. There's no single best imaging biomarker. The choice really depends entirely on the question the trial is asking. So the intended use of that biomarkers or that biomarker. If we get the use right, imaging really can become a quite powerful tool in trial design. So that's what we'll be focusing on today. We have the webinar structured around 4 central clinical trial applications, each of which represents a stage in a trial's life cycle where imaging can add different value. So we're looking separately at eligibility, enrichment, stratification, and efficacy application in clinical trials. And with this, I'm pleased to introduce our 2 speakers who will walk us through the imaging applications across those 4 areas. First, we'll have Martijn Müller, who is Professor of Translational Neuroscience at the Manchester Metropolitan University and who was formerly Senior Scientific Director for the Critical Path for Parkinson's CPP Consortium. And Martijn will take us through the PD trial landscape and the molecular imaging toolkit. So those markers coming from PET and SPECT across those 4 areas of application: eligibility, enrichment, stratification, and efficacy. Martijn will be followed by Kirsi, who's a principal biomarker scientist at IXICO. She's a senior clinical neuroscientist and chartered psychologist with over 20 years experience across Parkinsonian disorders and other CNS conditions. And Kirsi will show us how advanced MRI measurements can complement the molecular picture to look at differential diagnosis, stratification, and tracking of disease progression. Again, looking at those 4 different areas of clinical trial applications. And with that, over to you, Martijn.
Thank you very much, Robin. If we can go to the next slide, perhaps. Yeah, so I think Robin already covered this, that we will structure this particular webinar around these 4 different themes: eligibility, enrichment, stratification, and efficacy. If we can go to the next slide, please. So, nope, one back. So actually I want to, before I start, thank Robin for the very kind introduction and for the opportunity to present today. He actually already highlighted a couple of things that I wanted to say as well. But I think indeed that imaging has become an increasingly important component of Parkinson's disease drug development. I think it's really in a state now that it can complement traditional clinical outcome measures, and and really can provide insights into the underlying biology of the disease. I think at present, really no single imaging modality can serve every purpose across the clinical trial life cycle. So that's why we split it up in these 4 different themes, so to say. These different techniques really offer different windows into the disease process. So that is why in this webinar, we will view both nuclear imaging and MRI-based approaches. So can we go to the next slide, please? So Parkinson's disease is one of the fastest growing neurological disorders worldwide, and the number of affected individuals continues to rise globally. And in 2021, there were already 11.8 million people with Parkinson's disease. Despite the availability of symptomatic treatments, we still do not have an approved therapy that slows or halts disease progression. Next slide, please. I would say that there is hope. While challenges remain, there actually has never been more activity in the Parkinson's therapeutic pipeline. For the last few years, a group of Parkinson's research advocates and representatives from the Michael J. Fox Foundation and Cure Parkinson's have repeatedly come together to take an inventory of the drugs that are in the development pipeline. This group, led by Kevin McFarthing et al., showed that multiple approaches are being explored, including therapies targeting alpha-synuclein, lysosomal dysfunction, inflammation, mitochondrial pathways, and genetic forms of the disease. This creates both opportunity and urgency for improved biomarkers. If we can go to the next slide. Neuroimaging biomarkers for disease-modifying trials in Parkinson's will be mostly the focus of this webinar. Several challenges, however, continue to complicate Parkinson's disease-modifying trials. Disease progression is relatively slow, clinical measures can be noisy, and there's a substantial biological heterogeneity among patients. In addition, a big challenge is that distinguishing symptomatic effects from true disease modification. These challenges have increased the interest in objective biomarkers, including imaging. While several actually recent trials have missed their primary clinical endpoints, some actually have shown quite some encouraging signals in imaging secondary or exploratory endpoints. So it's certainly worthwhile pursuing the path of imaging. If we can go to the next slide, please. This need for biomarkers has been recognized by both the academic community and global health regulators. Biomarkers can improve selection, reduce variability, provide evidence of target engagement, and potentially offer earlier indications of efficacy. They are increasingly viewed as an essential component of modern Parkinson's disease drug development. And these academics, regulators, industry representatives have come together and developed complementary frameworks that were published, I think about 2 years ago. Next slide, please. Several imaging modalities are currently available, each providing different information about disease biology. Nuclear imaging techniques such as PET and SPECT can assess neurotransmitter systems, and brain metabolism, while MRI-based approaches can evaluate structural and functional changes. Routine clinical MRI is included actually in most trials, as these scanners are typically available at clinical trial sites, and the infrastructure is there. Nuclear imaging techniques typically require more specialized imaging centers. Overall, though, the maturity and regulatory acceptance of these methods vary considerably. Let's really start off now with a look at where imaging is already having the greatest impact in trials today. Let's go to the next slide. So I actually merged together eligibility and enrichment. Eligibility is the most universal application of neuroimaging, both for nuclear medicine techniques as well as MRI. And enrichment selects the right patients, typically those with active measurable disease in the target pathway. Next slide, please. The hallmark pathology underlying Parkinson's symptoms is degeneration of the nigrostriatal dopaminergic system driven by the deposition of alpha-synuclein in the substantia nigra pars compacta. By the time characteristic motor features of the disease emerge, substantial loss of dopamine terminals has already occurred, with estimates of 50 to 80% of the dopamine cells already lost. Because of this, though, imaging of this pathway provides a biological relevant target for confirming disease-related degeneration. Let's go to the next slide. That SPECT imaging, dopamine transporter SPECT imaging, assesses presynaptic dopamine transporter availability and has become quite an important diagnostic adjunct. It's currently approved to help differentiate Parkinsonian syndromes from conditions such as essential tremor, and is one of the few imaging biomarkers with broad clinical acceptance. In the left image, you actually can appreciate the notable difference in dopamine transporter density between a person with essential tremor in the middle of it, very similar compared to the normal control, and a person that has Parkinson's, which is in the right part of that image. For the person with Parkinson's, you can detect the pattern that is very characteristic. It's asymmetrical loss with the posterior part of the striatum, the putamen, more affected than the anterior part, the caudate. At the right image, you actually can show, or we actually can see differences in that SPECT binding across different stages of disease, where you already can appreciate considerable loss in the first panel of that figure, and it only gets worse over time. It also becomes more symmetrical. Let's go to the next slide. Importantly, for the drug development process, a proportion of individuals diagnosed clinically with Parkinson's disease actually do not show evidence of dopaminergic degeneration on imaging. These are commonly referred to as scans without evidence of dopaminergic deficit, or SWEDs for short. Inclusion of participants who have a clinical diagnosis and a clinical presentation that resembles Parkinson's disease, but are a SWED on scan, actually dilutes treatment effects in a clinical trial and increases the variability. So imaging really might provide a solution to identify those participants and either include them in the trial knowing that they are a SWET, or what happens more commonly, exclude them from the trial. Let's go to the next slide, please. Because of its ability to confirm dopaminergic deficit, dopamine transport or SPECT imaging has received support from regulatory agencies as an enrichment biomarkers for trials of early stages of Parkinson's disease. It helps ensure that participants have underlying neurodegenerative pathology consistent with Parkinson's disease. Collaborative efforts, such as the Critical Path for Parkinson's Consortium, can facilitate this regulatory process by bringing together all relevant stakeholders and encourage the sharing of data to make these kinds of endorsements possible. Let's go to the next slide. I'll talk now about stratification. While dopaminergic deficit is important, or confirming dopaminergic deficit is important, imaging can also help us understand the biological heterogeneity that exists within Parkinson's disease. As I mentioned, this brings us to stratification, which is, in this case, the process of identifying biological and meaningful subgroups within Parkinson's disease. Next. Although much of the focus thus far has been on Parkinson's disease as a dopaminergic disorder, the underlying biology is actually far more complex. Multiple neurotransmitter systems and pathological processes contribute to the symptoms and progression of the disease, actually creating a considerable variation between patients. Next slide, please. To address this variation between patients, subtyping frameworks, and there are many, Kirsi will show one as well, have been proposed using clinical, genetic, molecular, and imaging data. Many of these, I should note, of these frameworks actually have been determined in a data-driven manner. With this, the goal is to move beyond a single Parkinson's disease label toward biologically informed subgroups that may respond differently to specific therapies. Next slide, please. So if we move away from dopaminergic mechanisms, some of the non-dopaminergic mechanisms that may be particularly relevant for stratification are the cholinergic and the serotonergic system. Dysfunction in these systems actually contribute to symptoms that often respond quite poorly to dopamine replacement therapy. Imaging of these systems may help identify patients with distinct biological profiles and therapeutic needs. If you look at the left panel on this slide, there are actually multiple cholinergic PET imaging ligands available to look at the cholinergic system. But these are experimental ligands, and my colleagues here at Manchester Metropolitan University, in particular Nikki Ray, have also started looking at using MRI-based methodology to look at the cholinergic system, in particular using basal forebrain MR imaging. Serotonergic dysfunction is quite important as well, and can be looked at with DASB PET imaging, for example. Let's go to the next slide, where we will be focusing on the final use case in this case, and that is finding imaging markers that can provide evidence for efficacy of the drug intervention. Next slide, please. So we actually go back to dopamine imaging when we start talking about efficacy biomarkers. But in this case, I will not be talking about that SPECT imaging, but PET imaging. Here too in PET imaging, we have several approaches available for measuring dopaminergic integrity. Different tracers assess different aspects of dopamine function, including synthesis, transport, storage, and terminal integrity. Having the ability to look at these different constituents of the nigrostriatal pathway is critical actually in evaluating the effects of experimental therapies on the dopaminergic system. It's worth noting that currently of high interest are dopamine PET tracers such as PE2I and vesicular monoaminergic transporter type 2 tracers, VMAT2 tracers. I included an image here of VMAT2 DTPZ imaging, which is a carbon-11 tracer that I used in my studies when I was at the University of Michigan. But F18 versions are available of this as well. And there are several other efforts in the development of VMAT2 PET tracers. Let's go to the next slide. F-DOPA PET measures the dopamine synthesis and storage capacity and has a long history in Parkinson's disease research. It has been used in studies of restorative therapies such as dopaminergic cell transplantation, and it still remains to be one of the most established PET biomarkers in the field. Next slide, please. But challenges arise when you start looking at these different PET tracers, and one of these challenges in dopaminergic imaging has been the difficulty of comparing results across tracers and across studies. Recently, Roger Gunn and his group has proposed a centamine framework which seeks to harmonize quantification across dopaminergic imaging biomarkers, which really has the potential to improve the utility of dopamine imaging in multi-center clinical trials, even possibly using different dopamine PET tracers. Next slide, please. FDG PET provides actually a fundamentally different perspective by measuring brain metabolism rather than a specific neurotransmitter system. Using FDG PET, this actually has enabled us the identification of a disease-related metabolic network, such as the Parkinson's disease-related pattern, or PDRP for short, and the Parkinson's disease cognitive pattern, or PDCP. These network patterns are particularly interesting because they correlate with motor and cognitive manifestations of disease and have shown evidence of change over time. As a result, I would say that FDG PET network analysis could be explored as a candidate biomarker for monitoring disease progression and possibly for treatment response. Next slide, please. More recently, PET tracers targeting the synaptic vesicle protein 2A, SV2A, have enabled in vivo assessment of synaptic density. There's some recent studies on that, and these findings in Parkinson's disease, quite frankly, remain somewhat mixed. But having said that, these approaches still could provide an avenue for investigating synaptic dysfunction and neurodegeneration. Next slide, please. Given the central role of alpha-synuclein pathology in Parkinson's disease, an alpha-synuclein PET tracer is often viewed as one of the most important unmet needs in neuroimaging. Such a tracer could potentially provide direct evidence of target engagement for therapies designed to reduce or prevent alpha-synuclein aggregation. Significant progress has actually been made in recent years, and several candidate ligands have now entered human studies. However, no tracer has yet demonstrated the combination of sensitivity and selectivity needed for widespread adoption. Results have generally been more encouraging in multiple system atrophy, where alpha-synuclein pathology is abundant. Parkinson's disease and dementia with Lewy body remain more challenging because pathology is less dense and more heterogeneous. While alpha-synuclein PET is not ready yet for routine use in trials, it remains one of the most active areas of biomarker development and could eventually transform patient selection, target engagement, and efficacy evaluation. The imaging approaches we've discussed so far have been PET or SPECT based. Kirsi actually will now show you how MRI contributes across the same trial stages, beginning with eligibility assessment. With this, I'll hand it over to Kirsi.
Thank you, Martijn. So, using MRI for eligibility assessment can include excluding alternative diagnosis and identifying structural abnormalities or anatomical variants. Next slide, please. So, structural MRI is a key part of eligibility assessment. For example, MRI can identify vascular co-pathology, where more severe periventricular white matter or deep white matter hyperintensities, such as those you can see in image B of the top right picture of brain scans, those have been shown to be associated with non-motor symptoms in Parkinson's, including apathy, worse attention, working memory, and also processing speed. The table here shows the most routinely used structural sequences. There are others, such as F-Gator or what's called Bravo on GE scanners, and those can provide very detailed visualization of the basal ganglia area, which may sometimes be required for purposes such as surgical planning. It's always important to check, especially for trials that include neurosurgery, that deliver cell or gene therapy directly into the CNS, whether there are structural abnormalities or anatomical variants that could mean that the intervention is not suitable or safe. Even if considered safe, it might not be known how the finding could affect efficacy data. The picture on the bottom right shows an anatomical variant called cavum septum pellucidum, or CSP, which is a fluid-filled cavity visible on MRI as a midline CSF signal between the anterior horns of the lateral ventricles, which is highlighted there with the red arrows. It is generally considered a normal anatomical variant, but you can see that overall, structural MRI plays an important role in cohort selection, reducing confounds, and improving participant safety. Next slide, please. So now we're moving on to enrichment, and enrichment is really about selecting the right patients for your trial. Next slide, please. A modern screening pathway, exemplified here, would move us toward a more biological classification of Parkinson's disease. A clinical screen alone may yield a 5 to 15% SWED rate, so participants without that dopamine deficiency. Dopamine SPECT or PET helps to remove those. Further, alpha-synuclein seed amplification assay, SAA, or another way to detect pathologic alpha-synuclein, such as skin biopsies, adds to diagnostic confidence and helps to distinguish Parkinson's disease from multiple system atrophy, but it does not confirm Parkinson's disease with certainty. MRI, as the third step, can further enrich for participants with or without certain types of pathology. Structural MRI and volumetric analysis allows exclusion of those with atypical Parkinsonism-related signs and patterns of atrophy. For example, midbrain and superior cerebellar peduncle atrophy in progressive supranuclear palsy, or the hot crossband sign and cerebellar atrophy in multiple system atrophy, or parietal atrophy in corticobasal syndrome. A FLAIR scan enables quantification of white matter hyperintensity volume, where higher hyperintensity volumes have been found to be associated with worse progression of motor and non-motor symptoms, independently from age. A higher white matter hyperintensity burden may also mean higher risk of progression to Parkinson's disease dementia and to advanced disease stages in the next few years. Neuromelanin sensitive MRI, NMMRI, shows active nigral degeneration, which is particularly useful when the therapy targets the substantia nigra dopaminergic system directly. Adding free water diffusion MRI can help to identify those with high free water regionally, which can help to exclude those with neuroinflammation-related co-pathology, as well as to differentiate Parkinson's disease from atypical Parkinsonian disorders. Adding specific types of MRI to the screening pathway can add to the statistical power of the trial. The trade-off here between cost and patient burden versus statistical power depends on the trial phase, its size, and the therapy's mechanism of action. Next slide, please. With stratification, the key message is that although Parkinson's disease is biologically heterogeneous, imaging-defined subtypes exist and trials can pre-specify them. Next slide, please. So, why stratify? Well, different pathophysiological mechanisms underlie distinct subtype classifications, and this has implications for trials because treatment effects may be diluted, true efficacy signals may be masked, and larger sample sizes may be required to demonstrate efficacy. So how can MRI help here? Well, using MRI, we can identify distinct disease subtypes with different progression trajectories and potentially different treatment responses. This allows trial sponsors to prospectively define subgroups. Pre-specifying subgroup analysis by imaging-defined subtype allows the detection of treatment effects that in a pooled cohort would be masked. Next slide, please. So this example here includes 3 motor phenotype subtypes. A study by Ferres-Tenechard and others aimed to overcome previous limitations of Parkinson's disease subtyping by applying a data-driven clustering analysis to an extensive set of clinical measurements from a cohort of PD patients. This resulted in 3 distinct subtypes: mild motor predominant, intermediate, and diffuse malignant. Then Johansson and others validated the approach in a large longitudinal cohort of early to moderate Parkinson's disease, around 500 participants, by assessing the effect of the subtyping on clinical characteristics at baseline and during a 2-year follow-up. In that study, diffuse malignant patients compared to mild motor dominant patients showed involvement of more clinical domains, more diffuse hypokinetic rigid motor symptoms, and also faster progression. Different pathophysiological mechanisms focal versus more diffuse cerebral propagation may underlie the distinct subtype classifications. MRI provides objective biological evidence that clinically defined Parkinson's disease subtypes differ in underlying neurodegeneration and progression rates. The diffuse malignant subtype demonstrates greater baseline neurodegeneration and faster clinical and imaging progression than the mild motor predominant subtype. Stratifying patients by subtype may reduce biological heterogeneity and reveal treatment effects that could be obscured in a pooled analysis. The trial implications include that some patients may progress slowly, which can be helpful for prevention trials, but can be problematic when trying to detect disease modification, as well as the problem of ending up with a mixed cohort where effects may be diluted. By contrast, selecting participants with faster disease progression may increase sensitivity to detect disease-modifying effects. Next slide, please. Okay, now moving on to the final use case. So finding MRI endpoints that change measurably over time and reflect disease-related biology. Next slide, please. So starting with neuromelanin sensitive MRI, NMMRI, the neuromelanin contrast in the substantia nigra falls as dopaminergic neurons die. QSM, quantitative susceptibility mapping, quantifies iron in the substantia nigra, and iron rises measurably over time in Parkinson's disease. Free water in the posterior substantia nigra, in particular, has been identified a useful longitudinal progression marker in multiple cohorts. Combining the three gives orthogonal readouts of the same disease process. The same disease process. Then the last type of imaging I wanted to discuss is ASL. ASL is an MRI technique that offers a non-invasive way to obtain repeated measurements of cerebral blood flow for quantitative assessment of abnormal perfusion. It uses magnetically labeled water as an endogenous tracer. A Parkinson's pattern of decreased perfusion involves the basal ganglia, the parieto-occipital cortex, the precuneus and cuneus, and the middle frontal gyri. Studies have shown that ASL has the potential to predict motor progression in Parkinson's disease, and also that cerebral blood flow changes relatively little due to normal aging between the ages of 20 and around 80. Overall, advanced MRI provides quantitative readouts at a relatively low cost and good feasibility for repeated measurements. The three key efficacy sequences, neuromelanin MRI, QSM, and diffusion MRI, all fit inside about 30 minutes of scanning, and they do not require any radiation exposure beyond the magnet. With ASL, all major MRI platforms have ASL sequences capable of obtaining high-quality whole brain perfusion images within a few minutes of scanning. The images here, if I go through those, the neuromelanin images at the top, they are at the level of the substantia nigra for a healthy adult, a 65-year-old male, and a patient with Parkinson's disease, a 71-year-old male. And you can appreciate the reduced hyperintense signal in the substantia nigra of the patient relative to the control. Next, the QSM images show representative images of healthy controls and participants with early Parkinson's disease. The Parkinson's disease QSM images show brighter signals in the midbrain and basal ganglia areas than the healthy control images, which demonstrates higher iron accumulation in Parkinson's disease. Then at the bottom, the free water diffusion MRI images show midbrain with the free water data for one healthy control participant and one participant with Parkinson's. The participant with Parkinson's has higher free water values in the substantia nigra, especially in the posterior part, which is shown here in the blue color, indicating more free water. And the last image, the ASL versus FDG PET image, shows transverse ASL and FDG scans of a healthy individual, male, aged 57, with normal mini-mental state examination. And this shows how FDG PET and ASL do not show exactly identical, but quite similar data in terms of where there is higher and lower glucose metabolism in FDG PET and lower cerebral blood flow in the ASL image. And because of this coupling between brain metabolism and perfusion, ASL could be considered as an MRI alternative for FDG PET, especially in studies that need SPECT or other types of PET, and the inclusion of FDG PET would be the only reason to use sites with PET or PET-MR scanners instead of sites with MRI only. In addition to lower costs, the choice of ASL could mean lower patient burden. As with other types of advanced imaging, though, the use of ASL in multi-center studies requires consideration during trial design and setup for how to ensure consistent and usable data across all sites and scanners. And that's it from me. So we can proceed to the next slide. Thank you.
Sorry, need to put my microphone back on and my screen. There I am. Thank you very much, Kirsi, for this great overview of MR-related methodologies that can be used in the drug development cycle. I actually think that personally, I think that there's a lot more enthusiasm about that, especially given the findings that were presented at some of the recent conferences regarding clinical trials that showed effects of the drug on MR-based measures, as I had mentioned before. But I hope it has become clear that imaging can contribute across the entire clinical trial pathway, from confirming eligibility and enriching cohorts through biological stratification to providing quantitative measures of efficacy. I do want to end with, next slide please, with a forward-looking slide regarding some of the future directions and conclusions. So looking ahead, I do think that imaging biomarkers are unlikely to replace clinical outcomes, but they will increasingly complement them. The future really lies in integrating imaging with fluid biomarkers, genetics, and digital measures for that matter, to create more biologically informed clinical trials and support precision medicine approaches. Every advance in imaging brings us closer to more efficient clinical trials and ultimately to therapies that can slow or prevent progression. Progression in this field actually can only be made by sharing of data and knowledge. For people living with Parkinson's today, that sense of progress is a reason for genuine hope. And with that, I think we'll hand it over to Robin for some questions and answers.
Great. Thanks, Martijn. Thanks, Kirsi. Great presentation and so much going on in this space, so many things happening. And we have time, which is great because we have think we have a quite interactive audience with various questions coming through already. Actually, I'll kick it off though with one question from my side, just a general question, as we have seen so many different modalities and techniques, and so maybe to Martijn, if I'm a sponsor planning a PD trial now, and maybe I'm new to imaging, what would you say is the single most important question I should keep in mind before choosing any of those modalities?
So I think the most important question is, what is your intended use? What is it that you want to show? What is your research question, so to say? I guess intended use is perhaps the regulatory term for that, and I don't even want to go to the context of use just yet. But I think what we saw with these different use cases is that for each stage in the drug development process, for each of these different questions, there's actually different modalities, different technologies, different tracers, perhaps, that can answer these questions. There's unfortunately at this moment no magic bullet, no one imaging thing that you can do that would address all of it, right? So I think right now, if you're just interested in, how can I have an optimum group of Parkinson's disease patients, making sure that they all truly have Parkinson's, then I would go with MRI, perhaps T1, just to see if there's some kind of form of vascular Parkinsonism, stroke, and things like that, and a dead SPECT scan, just to confirm the dopaminergic deficit. So that addresses that research question. Are you interested in stratification? Then it becomes a whole different question, whole different set of technologies that are available to you. So intended use is the most important thing. Really ask yourself, what is it that I want to show? What is it that I want to address? Secondary to that, I think it comes to things like maturity, which we show the maturity of the methodology. Has it been tested in just one center? Have different centers looked at it? Where are we in terms of validation? So I think that I hope that answers part of your question.
Yeah, that's great. And actually, I think some of what you said links to a question we got from the audience, which is, how do you determine whether an imaging biomarker is fit for purpose for a particular trial? I think you've touched on some of this, but I'll put the question out there anyway. If there's anything you want to add, Martijn, or maybe Kirsi, you want to add to that question.
Yeah, so it pretty much goes back to what I was saying. Indeed, don't come to people like IXICO and say, I need something good that will help me for the clinical trial, because I don't think that is going to help you. You need to come with a very specific question, saying, I need help with enrichment of my trial, or eligibility of my trial. So that's where you start. Again, back to the intended use. But then it really becomes important. This is when you really start moving to fit for purpose, when you start addressing some of these validation issues, like has it been done in different centers? If you look at different methodologies, do they tell the same story? Different analytical approaches, do those tell you the same story? And so that's a long list of validation that you have to go through for something to become really fit for purpose. But your research question, I think, is the one to start with. Validation is the next one that that you need to address before you can start implementing it with some confidence in your trial.
That makes sense, and I think we'll come back to that maybe in a little bit, because there's one operational question that probably touches us back to that topic. But I want to maybe get into some of the MRI and PET questions that came through on the chat. Maybe the next one for Kirsi. Where does advanced MRI add something that molecular imaging can't, especially for telling PD apart from other Parkinsonian disorders?
Yeah, so hopefully I managed to address some of that in my presentation. For example, when I talked about enrichment, so it's now thought that these advanced MRI sequences could help to differentiate between Parkinson's and atypical Parkinsonism, for example, between Parkinson's and MSA or Parkinson's and PSP, and especially free water diffusion MRI could be used for that, to look at free water measures from multiple regions, not only the substantia nigra. And then other things, I think MRI can contribute to that enrichment part in many ways, as I mentioned, for example, to look at the white matter hyperintensities, and then you can include or exclude participants with a certain number or certain volume of those. Then efficacy, I think we're still not using PET so much for efficacy. SPECT has been used a little. So MRI has more types of sequences, I think, that you can use, at least for exploratory efficacy analysis.
Great, thanks. There's actually two questions maybe for Martijn on dopamine and associated biomarkers. First one, what imaging methods do you think would be most useful for sweats, as the underlying pathology in these subjects is likely to diverge from non-sweat? So that's a bit more conceptual one.
So I'm not sure if I fully understand the question. So for identifying sweats, you would use dopamine transporter imaging, for example.
Well, this is not how I read the question, and obviously that's what you've explained. But the way I read the question is, so we have already established there's no dopamine deficiency in those subjects. What imaging techniques could we use to better understand the clinical symptoms that maybe we've seen on those subjects that we couldn't confirm through dopamine deficiency, if that makes sense?
Okay.
At least that's in my mind an interesting question. I apologize to the question, to the person who asked it, if that's not the intended question, but that's how I read it.
Okay, yeah. So that's interesting in itself, right? So when you look at clinical trials, and the numbers vary, but also when you look at cohort studies, there's a considerable percentage of sweats that are being identified, especially in these early stages of Parkinson's disease. So numbers vary. I think a good estimate is probably of all the dopamine DET scans that you do, that about 15 to 20% is a sweat. But then the question is, they still, have Parkinsonian symptoms. They have been diagnosed clinically with Parkinson's disease. So what is going on, right? So in that case, the MRI actually might give some explanation to that. There might be strokes, things, tumors, even perhaps things that can be identified on an MRI. But then, what is the fate of sweats? That's actually not that well known. I would say, there's really, as far as I know, really very little, maybe perhaps none, follow-up studies that follow sweats, consistently over time and do imaging on them that might explain, what gives them Parkinsonian symptoms. I have heard that some of the sweats actually do convert to Parkinson's disease later on. So it just might be that the DET scan didn't pick that up just yet, and that you're very early in disease. But I do think indeed that follow-up is needed, and that imaging might help with that. But I actually don't have a good answer as to what would make a sweat, and what the profile would look like if you just keep consistently following them over time.
Yeah, I think if I can maybe play that back in a brief summary, what I heard is certainly looking at an MRI is useful because you can see whether there's co-pathology that may explain some of the just the clinical symptoms that we've seen. So we're actually coming up close to the hour. There's two questions I still want to ask, so maybe asking you for a very brief response. Martijn again, so the question here is, that SPECT is obviously well established to look at dopamine deficiency. You touched on dopamine PET imaging as well. So can you maybe briefly comment on what the right tool for which application is? Sorry,
what the right tool for?
How would I choose? So the question that came in here is where is that SPECT still the right tool, and where should sponsors be looking at PET instead? That's a question.
Okay, yeah, no, that's a very good question, and so I think it's about scalability, right? So I think for the PET tracers, the ones that I mentioned, PE2I, and the VMAT tracers, they're still in an experimental phase. So not everybody is able to synthesize these tracers. You need a cyclotron. And typically you need to do that at the more specialized imaging centers. So that typically would really restrict your use of those tracers in your drug development process, unless you have a very particular question to that. I would say go with that SPECT, which is much more scalable, readily available. In many clinical trial sites, and many hospitals have the possibility and have the means to actually do those types of scans. So again, it comes a little bit back to your question. If you have a very specific research question that would need a PET tracer. But otherwise, you also need to start looking at things like scalability, cost, and things like that in making your decision.
That's great. Thanks, Martijn. I really like to keep going, but unfortunately we are at the end of the webinar. So we will follow up with individuals if there are questions that we haven't answered yet. We'll follow up by email. And that leaves me to thank both speakers again and hand it back to James for some final housekeeping notes.
Thanks, Robin. So thanks all again. Thanks for the speakers. As Robin said, if you have any additional questions, we can cover those on email. Please email us directly with those. And we will have captured all of the questions, and we will obviously publish the webinar, and so you'll have access to this digitally probably in the next 24 hours. So if you'd like to revisit or refer other people to it, please do so. As I said at the beginning, this is the first of this is the third of a series of 6 we're doing. So please do watch out. We will be publicising further webinars in AD, PD, and some technology and AI use in imaging over the next few months. So watch this space. But thank you all for joining.
Thank you very much.
Thank you.
Thank you. Bye-bye.